Alright, next we're going to be looking at neurotransmitters, which are the language of the nervous system. There's about 50 or more that have been identified. But most neurons make 2 or more neurotransmitters, and they can exert several influences depending on the receptor and postsynaptic membrane that they bind to. Usually we see neurotransmitters released at different stimulation frequencies, and so they're classified by chemical structure and by function. So let's start by looking at one you should be familiar with, which is acetylcholine. Now, it was first identified and it's the best understood. It's released at neuromuscular junctions, but also by some autonomic nervous system neurons and some central nervous system neurons. It's synthesized from acetic acid and choline, and that's going to be synthesized by the enzyme choline acetyltransferase. And we should already know that it's degraded by the enzyme acetylcholinesterase, where part of it can be reused at the axon terminal. So when we look at this neurotransmitter, we're essentially looking at one that's almost always excitatory, but is especially always excitatory on skeletal muscle cells. Now we can also look at biogenic amines like the catecholamines, dopamine, norepinephrine, epinephrine. These are synthesized from the amino acid tyrosine. There's also the indolamines like serotonin and histamine, and serotonin is synthesized from the amino acid tryptophan, while histamine is synthesized from the amino acid histidine. And all of these biogenic amines, the catecholamines, the indolamines, they are broadly distributed in the brain, and they play behavioral roles, especially when it comes to emotional regulation and your biological clock. Now, some autonomic nervous system motor neurons also relay or secrete norepinephrine. And oftentimes we associate imbalances with these neurotransmitters with mental illness. For example, overactive dopamine signaling occurs in schizophrenia. But you might also know of norepinephrine and epinephrine, the catecholamines, as adrenaline. They can be produced by your adrenal medulla and secreted directly into the blood as hormones in that case. We also have amino acids, which are just glutamate, aspartate, glycine, and a derivative of gamma aminobutyric acid that are found only in the central nervous system and function mainly among those interneurons. Now, slightly longer neurotransmitters, so a couple of amino acids bound together, the neuropeptides or peptides, include a substance P, endorphins, and gut-brain peptides. Substance P is a mediator of pain signals, while the endorphins like beta-endorphin, dynorphin, and enkephalins are going to act as natural opiates to reduce pain perception. They actually bind to the same receptors as opiates and morphine. Then your gut-brain peptides like somatostatin and CCK called cholecystokinin, those are going to be helpful in communicating to your GI tract. We also have purines, which basically is ATP. It includes a lot of adenosine. It's a great inhibitor in the brain or with central nervous system neurons. Caffeine stimulatory effects are a response to the blockage of adenosine receptors, and this can act in both the central nervous system and peripheral nervous system. And this can produce fast or slow responses. It all has to do with a lot of induction by calcium influx via astrocytes. But let's not forget gases and lipids, or the gasotransmitters. The main one's going to be nitric oxide, but there's also carbon monoxide and hydrogen sulfide gases. They all bind to G protein coupled receptors in the brain, which initiate second messenger systems, but they're also all lipid soluble. We don't store a lot of them, we don't keep a lot of them on demand. They have to be synthesized when they're needed. We're going to focus mainly on nitric oxide because it's involved in learning and formation of new memories, especially as it's present in the central nervous system. But in the body or in the peripheral nervous system, and especially if it makes it into the blood, it's a potent vasodilator. So you don't want too much of it, which is why it's only synthesized on demand, because excess can lead to brain damage in stroke patients, but it can also cause smooth muscle relaxation in the intestines. So it's not all bad, but it has to be regulated. And then when it comes to hydrogen sulfide gas, it can act directly on ion channels to alter function. So again, we want to modulate and synthesize and regulate these gases and lipids because they have such potent functions. And then last but not least, at least chemically, we're going to look at the endocannabinoids, which act on the same receptors as THC, which is the active ingredient in marijuana. They also are going to be common in G protein linked receptors of the brain. and they're also lipid soluble, so they're kind of similar to the gasotransmitters. They are also synthesized on demand, but they are believed to be involved in learning and memory, but we do know they also may be involved in neuronal development, controlling appetite, and also suppressing nausea. So that's looking at kind of the structural classification, or looking at what are the neurotransmitters made of. Are they made up of peptides or just an amino acid? Is it acetylcholine, which is kind of unique, or is it a lipid or a gas? So that was the structural classification that we just looked at. But we also want to look at the functional classification of neurotransmitters. So we want to look at what do they do. When it comes to the functional classification, there are two types, excitatory and inhibitory. Excitatory neurotransmitters cause depolarizations, whereas inhibitory neurotransmitters cause hyperpolarizations. You've been introduced to that idea already. Depolarizations are excitatory. Hyperpolarizations are inhibitory. So what we would call an excitatory signal that reaches a postsynaptic cell is an EPSP, or excitatory postsynaptic potential. What we would call an inhibitory signal that reaches a postsynaptic cell is an IPSP, or an inhibitory postsynaptic potential. So some neurotransmitters can be excitatory and inhibitory. Others are only one or the other. A great example, though, is looking at acetylcholine. Its action is determined by its receptor type on the postsynaptic neuron. Because acetylcholine is always excitatory at neuromuscular junctions, or when we're looking at skeletal muscle. But acetylcholine is inhibitory when it binds to receptors on cardiac muscle. So we can see that neurotransmitters can sometimes do both, be excitatory and inhibitory. All right, next we're going to move on and look at neuronal circuits. Now these are patterns of synaptic connections in neuronal pools, and they're called circuits because they determine the pool's functional capabilities in a series of neuronal communication. The first neuronal circuit we're going to look at is a divergent neuronal circuit. It's where we have one incoming fiber, then it stimulates an increasing number of fibers, and so it's going to increase via an amplifying circuit. Divergence can occur along a single pathway or several, but they're always known as amplifying circuits. There's also the opposite of divergent circuits, or convergent circuits, where you have a strong stimulation or strong inhibition that acts on one output neuron, creating a funneling or concentrating effect. This is really common in sensory and motor pathways. But we can't forget the reverberating or oscillating collateral synapses that combine essentially positive feedback mechanisms in a neuronal circuit. So with this, we're going to have previous neurons in a chain communicate back to prior neurons in that chain. This is essentially how a positive feedback mechanism works. And as a result of the positive feedback, the impulses reverberate or are sent through the circuit again and again, giving a continuous output signal until one neuron in the circuit fails to fire and turns off the positive feedback mechanism. So we see this a lot with control of rhythmic activities like the sleep-wake cycle, breathing, certain motor activities such as swinging, or something like moving your arms when you walk. That's when we would see reverberating or oscillating neuronal circuits. Then we have parallel after discharge. Now with this, incoming neurons stimulate several neurons in parallel pathways or arrays that eventually stimulate a common output cell. Now the impulses would reach the output cell at different times, so that would create a burst of impulses called an after discharge that can last 15 milliseconds or more after the initial input has even ended. So this is not positive feedback because once all the neurons have fired, the circuit activity ends. So we might see this more in something like complex exacting types of mental processing. So really, really common actually in the central nervous system where we have interneuron association and integration. And then our last topic to look at is just a quick look at a homeostatic imbalance, which is the breakdown of myelin sheaths in a disorder called multiple sclerosis. This is an autoimmune disease that affects primarily young adults, but this is when the myelin sheath in the central nervous system is destroyed because the immune system is attacking the myelin. What happens to the myelin is it turns into hardened lesions called scleroses, and that's going to slow impulse conduction and eventually cause the ceasing of impulse conduction. The demyelinated axons do initially increase sodium channels, and that causes kind of moments of remission for those with multiple sclerosis, but those individuals generally do relapse. Symptoms are widespread: visual disturbances, weakness, loss of muscular control, speech disturbances, and even urinary incontinence. What's the treatment though? Well, drugs that modify the immune system's activity can improve the lives of those with multiple sclerosis, but it is a condition that is progressive, and we have no cure. So the prevention is really kind of the best bet for those who are concerned, and we've found that high blood levels of vitamin D can reduce the risk of development, but they don't ensure that it will not develop. So here we can see an image on the left where we can see the degeneration of the myelin. When that hardens, it forms the scleroses. When you have multiple, that is where we're getting the name multiple sclerosis. The images on the right are showing a normal versus a damaged myelin sheath in the central nervous system. So keep in mind that this is most common in the central nervous system, but it doesn't mean that the peripheral nervous system cannot be later affected. Alright, that is actually it for Chapter 11, which is your introduction to the nervous system. If you haven't already, make sure to watch all parts.